Hello, my name is Trevor, and I'm a PhD student at Harvard Medical School. I work in the Gallenborg lab and develop interactive visualization tools for exploring spatial, omic, and microscopy data. Today, I'll be sharing Viv, a web-based tool for building interactive visualizations for primary high-resolution microscopy images. Advances in single-cell technologies and highly multiplexed imaging have enabled the comprehensive profiling of complex tissues in diseased and healthy states. Associate efforts like the Human Cell Atlas and NIH HubMap are some of the largest producers of these integrated data sets, and our lab is a part of the HubNet consortium. A primary goal of ours is to develop infrastructure to help share and explore these integrated data sets. We want to empower end users to both browse, explore, and analyze these data with ease. Today, I'll be talking about some of our work with high-resolution microscopy data and thinking about how to design these data portal client applications through our web-based technologies. So to reiterate, our goal is to think about simplifying this computational infrastructure so that data providers can focus on sharing the data, and then we can start to build computational tools that can interface with those data portals and provide easy access to end users. Before I talk about Viv, I first want to put the work into context by describing at a high level what needs to occur to render multiplexed and multiscale imaging data on the web. Primary data produced by multiplexed imaging methods are typically represented as multidimensional image stacks, where each channel is typically some sort of chemical signal. These data can be stored in the image format shown here on the left. It's important to note that these files may contain tens or hundreds of different channels depending on the acquisition method. Each of these channels is captured with, may be captured with 16 or 32-bit precision, and therefore must be pseudocolorized in order for some sort of visualization. So ultimately, there's this data transformation that needs to occur that takes these blue data tiles denoted here on the left and ultimately creates the pixels that we end up seeing on the screen. And today what I want to describe is how Viv differs from typical web-based viewers in the way in which it combines and renders these channels. The first approach is offline server-side rendering. In this method, all data transformations are decided before deploying to a web application. Channel groupings, colorization, and contrast limits are all decided beforehand, and a pre-rendered version of the dataset is generated once on the server. This method is good for presentation, but ultimately a complete copy of the imaging data is created, and these rendered image tiles are not useful for analysis. In addition, a user cannot redefine any type of data transformation, so they must download the complete data in order to explore it. The next approach is online server-side rendering. In this method, a sophisticated server is set between the data and the client. The server is responsible for dynamically reading and rendering data tiles according to user-defined transformations. While this approach allows the user to change the visualization, any changes to the rendering require a server round trip, which can be expensive over a network. In addition, the architecture requires maintenance on both the server and client that can be difficult to maintain. A slightly nuanced point is that the server and client are very tightly coupled in applications with this architecture. This can make it difficult to reuse the client web application in a different context. The final approach is client-side image rendering. This method mirrors that of desktop applications where the client application alone interfaces with the dataset. In client-side rendering, raw data tiles are fetched and rendered purely by the client. Once the data tiles are loaded, subsequent re-renders are very inexpensive. This approach decouples the client from the server since all transformations are defined and applied within the client application. The approaches are shown here for comparison. As you can see, the client-side applications for both server-side approaches rely on data to be pre-rendered into image tiles, meaning that clients cannot interface with the primary imaging data directly and instead must go through a server. Viv is unique from other web-based applications because it employs purely client-side image rendering. Data tiles are loaded dynamically directly from the image data and are rendered on the GPU on the client. This architecture makes Viv flexible to build applications with. We have packaged Viv as an open-source JavaScript library that can be installed via npm. The library is a toolkit for building interactive visualizations for primary multiplexed and multiscale imaging data on the web. Viv's data loaders are responsible for fetching data tiles directly from open standard OMI-TIFF and OMI-ZAR formats via HTTP. We have worked closely with the Open Microscopy Environment team to build support for these open standards for sharing bioimaging data. The Bioformats tool suite and software can be used as a mechanism to convert primary imaging data from vendor formats into one of these open standard compatible formats. The rendering component of Viv are packaged as DeckGL layers. The layers are a fundamental building block in the DeckGL ecosystem, which is a high-performance WebGL framework. And these layers can be composed back with other layers from DeckGL to create more advanced multimodal visualizations. Viv is intentionally modular by design and can thus be reused in a variety of custom applications that choose to include multiplexed imaging data. Avivator is a standalone web viewer that supports viewing OMI-TIFF and vizarr data formats via URL. It's developed closely alongside Viv, the library. It is versatile and highlights many of the exciting features that we've recently added to Viv, and Avivator includes features like picture-in-picture views, side-by-side views, local lens transformations, and support for 3D visualization. The magic lens in Avivator re-render the image with different types of transformation settings. For example, a lens can be used to highlight a particular channel in the source image. Since all operations take place on the client, there's no noticeable lag when moving the lens over the image. vizarr is a minimal viewer for vizarr-based images only. It aims to support the developing OMI-vizarr specification for the open microscopy environment. We integrated vizarr in Jupyter Notebooks as an ImJoy plugin, meaning that vizarr can be used as a front end to different types of remote computational environments. This Google Colab notebook shows vizarr usage in this context. The Mandelbrot set is implemented as a generative multiscale vizarr store in Python, and the vizarr client allows us to interactively view this source, which is in the Python backend. This example demonstrates the flexibility and integration of vizarr into a different type of computational environment. In our paper, we describe a novel remote image registration workflow powered by vizarr. Multiple registration methods are tested in a remote high-performance cluster system, shown here on the right, in a Jupyter kernel. The results of these registration methods can be viewed in the full resolution within the vizarr client, all on someone's laptop. The sampling of registration methods is then delegated to this high-performance system, while a user is able to interact with the system via the same computational environment and view the same results in the same notebook. In addition to usage in Jupyter, vizarr can be used as a standalone viewer and access vizarr datasets via URL. Several datasets from the image data resource have been converted to the OMI-vizarr format and exposed for public consumption on cloud storage. Since the viewer only requires a data URL, this archive can be viewed directly in the browser with vizarr, or another non-web client like Dapari that understands this data URL. In this example, I have opened the OMI blog website, which displays several of these different datasets that have been converted from the IDR. I'm able to open vizarr in a new tab, and then also copy the link to the URL and open the same image dataset in Dapari. This example demonstrates the ability to share datasets between different users and tools via data URL. Thank you very much for your attention, and I look forward to your questions.